A good day to everyone. For this video lecture, I'm going to talk about I'm going to talk, to continue to talk about the cell division, specifically on the mitosis and the meiosis. So, last video lecture, I've talked about the interface. I've talked about the parts of the interface, and I've talked about what will it do on the DNA and what will it do on the chromosome. So mga ganun. So for this video lecture, I'm going to talk about first on the mitosis. So ano nga ba yung mga parts niya? Ano nga ba yung mga process na gagawin niya? So as an introduction muna on the word na mitosis, it means threads. So as you can see on the figure, parang itong mga threads na to. So yun yung it comes from the word yung mitosis, it comes from the word threads, and it refers to the thread-like appearance of the chromosome as the cell prepares to divide. So yun. Kasi parang threads daw yung chromosome pag magdi-divide na siya. So, yun yung doon nila kinuha yung word na mitosis. So, on the early parang stages ng mga study nila, yung mga microscopists, they were the first to observe these structures, and they also noted the appearance of a specialized network of microtubule during mitosis. So sinasabi yung mga early microscopists, yung mga scientists, sila daw yung first na nag-observe nitong appearance ng thread-like appearance ng chromosome during yung pagdi-divide nila. And then they also show yung specialized network of microtubules. So as you can see in the picture, itong mga microtubules na to, nakita rin nila during yung mga early stages or early part ng pagsi-study nila noon. So, mitosis is a type of cell division in which one cell, which is the mother, divides to produce two new cells, the daughters, that are genetically identical to itself. So, on the picture you can see, so you can imagine this as the mother or the cell. Now, there are processes na magaganap hanggang doon sa dito sa part na to, dito sa last part, wherein it produces two new cells. Ito yung mga daughter that are genetically identical to itself. So, bakit nga ba genetically identical to itself? Because it comes from yung iisa, yung sa mother mismo. Nag-divide lang siya into two new daughter cells that are genetically identical to itself. So, in context, yung mitosis, yun yung part of the cell division process in which yung DNA of the cell's nucleus is split into two equal sets of chromosomes. So, ito. So, yung majority of cell division that happens in our body involve yung mitosis, yung pagdi-divide from one cell into two daughter cell. Yun yung mostly yung majority ng cell division, yun yung nagaganap mitosis doon sa body natin. So, the goal of mitosis is to make sure that each daughter cell gets a perfect full set of chromosome. So, yun yung goal ng, one of parang goal of the mitosis is to produce yung mga daughter cells that have perfect full set of chromosome. So, yung mga cells that are too few or too many chromosomes, yung mga na-produce na mga ganun, yung masyadong maliit yung pair ng set ng chromosome or masyado namang malaki or marami yung set ng chromosome, they do not function well. Meaning they may not survive or itong mga chromosomes na to, yung too many chromosome or too little chromosome na produce doon sa daughter cell, it can also cause yung cancer. So, when yung cell undergo mitosis, uh, hindi lang sila nagdi-divide, hindi lang sila dinidivide yung DNA nila at random. Pero itong um, mitosis na to, they will split up and they duplicate yung chromosome in a carefully organized series of steps. So, may mga sinusunod na series of steps sa mitosis para maiwasan yung pag-produce ng daughter cells that have too few or too many na chromosome. So, ano nga ba itong mga series of steps na to? So, you have here yung steps or phases of mitosis. So, you have prophase, and then you have metaphase, and then you have anaphase, and then last is telophase. Now, itong cytokinesis, I will discuss this further on. Pero it may happen during sa anaphase part or doon sa telophase part, itong cytokinesis. So, basta the phases of the mitosis, it involves itong apat na to. So, in order to parang memorize itong phases of mitosis, para madaling ma-memorize itong phases of mitosis, yung parang ginagawa ko is tinitagnan ko itong first part. So, PMAT. So, P stands for prophase, M stands for metaphase, A stands for anaphase, and then T stands for telophase. So, yun, yung ginagawa ko. Basta, pag phases of mitosis, dapat yung unang papasok sa isip nyo is yung PMAT. PMAT, prophase, metaphase, anaphase, telophase. So, first, let's discuss about the prophase, specifically on the early prophase. So, in early prophase, the chromosome start to condense, making them easier to pull apart later on. So, may stages doon sa mitosis kung saan ipupull apart mo yung mga chromosome. So, in early prophase, yung ginagawa is kinocondense yung mga chromosome. Hindi sila yung parang naghihiwalay-hiwalay. So, nagko-condense sila yung mga chromosome. And then, another one is yung mitotic spindle begins to form. So, itong mitotic spindle, ito yung parang structures of microtubules. Ito ay mga strong fibers that are part of the cell's skeleton na tinatawag. So, yung ginagawa ng mitotic spindle is to organize the chromosomes and move them around during mitosis. So, itong mitotic spindle, sila yung responsable para gumalaw at pull apart yung mga chromosome during mitosis. So, itong spindle na to, it grows between the centromeres as they move apart. So, another one is yung nucleolus or the nuclei. A part of the nucleus where ribosome are made disappears. So, itong part na to, nucleolus or yung nucleoli, it will disappear during the early prophase. So, yung pag-disappear ng part na to, it is a sign that the nucleus is getting ready to break down. Kasi itong nucleolus, it is part of the nucleus. So, pag nag-disappear na tong nucleolus, it is a sign that the nucleus will, it is getting ready to break down also. So here, we have here the figure doon sa early prophase na tinatawag. So, ito yung laging tatandaan as you can, ito palang presentation, it is lifted on a site called Khan Academy. So, they actually, maganda yung mga parang summarization nila. It is simple enough that we can understand it. And then maganda rin itong mga figure na ginagamit nila. So, in early prophase, yung laging tatandaan, first, yung chromosome starts to condense. So, yun, parang nagko-condense sila sa isang part. And then, yung nucleolus is gone. So, wala na yung nucleolus. And then, yung mitotic spindle starts to form. So, yun, parang yan. Yung mitotic spindle, yun yung mga microtubules. So, importante to during the later part of the mitosis. So, yun yung early prophase na tinatawag. Now, for the late prophase naman, yung microtubules can bind to the chromosomes at the kinetochore na tinatawag, which is a patch of protein found on the centromere of each sister chromatid. So, I hope that you remember pa rin yung parang structure ng chromosome. Structure ng yung chromosome wherein doon sa middle kung saan nag-bind yung dalawang chromatids, yung sister chromatid, nandoon yung centromere, di ba? So, yung microtubules ngayon, parang magbabind doon sa centromere, specifically doon sa kinetochore na tinatawag, which is a patch of protein that is found on the centromere. So, basically, the microtubules will bind with the centromere on that is located on the sister chromatids doon sa parang gitna, let's just say gitna ng dalawang chromatids. Now, the microtubules that bind a chromosome are called kinetochore microtubules. So, kung nag-bind na yung mga microtubules, it will become kinetochore microtubules na tinatawag. So, ito, di ba, pinakita ko kanina sa early stage ng itong structure na to. So, nabubuo na ito, itong spindle na tinatawag. So, itong part ng spindle, you have yung anong tawag nito? Itong mga microtubules na tinatawag. So, itong microtubules, you focus on the gray na parang line. Ganito yung mangyayari. Yung gray na line, magbabind doon sa centromere. Specifically on its kinetochore. So, yun yung gagawin sa late prophase. Yung mga microtubules magbabind doon sa centromere, specifically on the kinetochore. Now, pag nagbind na yung microtubules with the kinetochore, magiging kinetochore microtubules ka na. As you can see on the figure, nagbind yung microtubules doon sa kinetochore, doon sa centromere ng mga chromatids. So, as you can see also on the picture, may mga parang microtubules that did not bind with the kinetochore, na did not bind doon sa centromere. So, yung mga microtubules that don't bind to the kinetochore, pwede silang mag-bind or mag-grab doon sa microtubules on the opposite side. So, parang ganito sa part na to. Magbabind sila doon sa microtubules on the opposite side. Pag nag-bind na sila, it will stabilize yung spindle. Itong structure na to. So, yun yung mangyayari sa late prophase. Basta yung tatandaan, yung mga microtubules, they will bind into the kinetochore doon sa centromere. And then yung mga microtubules na hindi nag-bind doon sa centromere or sa kinetochore, it will bind with the opposite microtubules and thus it will stabilize itong spindle na tinatawag natin. So, again, in the late prophase pa rin, the mitotic spindle now begins to capture and organize the chromosome. Di ba? Yung mitotic spindle wherein nandun yung microtubules, nagbabind na sila doon sa mga chromosome, specifically on its centromere. So, with that, yung mitotic spindle now, it will begin to capture it kasi nakabind na mismo. it will begin to capture and organize the chromosomes. So the chromosomes become even more condensed so that they are very compact na. So hindi na sila nag-scatter, nagko-condense at nagko-condense na sila. So the nuclear envelope will break down, releasing the chromosome. So with that, malaya na yung mitotic spindle to organize yung mga chromosome. Kasi yung parang nuclear envelope na tinatawag, it will break down. Kasi itong nuclear envelope na to, parang it will hold in place yung mga chromosome. Pero dahil sa late prophase, dahil nag-break down yung nuclear envelope, it can enable yung mitotic spindle to capture and organize this chromosome. Now, the mitotic spindle will grow more and some of the microtubules start to capture yung chromosome na tinatawag. So here, this is the figure kung saan. So by the way, yung late prophase, pwede rin tawagin na pro-metaphase. Tinatawag rin na pro-metaphase. So yun, yung nuclear envelope breaks down and then chromosomes will fully condense and then itong mitotic spindle will begin to capture and organize itong mga chromosome. Bakit nga ba niya i-capture at i-organize? Kasi on the third part or on the second part pala ng mitosis which is metaphase, the spindle na-capture na niya yung mga chromosome and it will line them up at the middle of the cell ready to divide. So yun yung purpose kung bakit hinuhuli or kinakapture ng mitotic spindle yung mga chromosome para i-line up niya ng maayos para maganda yung process ng pag-divide nila. So all the chromosomes will align at the metaphase plate. So itong metaphase plate na tinatawag, hindi ito parang physical structure mismo, pero itong metaphase plate, it is only a term that will parang sinasabi ng mga scientists to connote yung plane where the chromosomes line up. It is only a parang term na sinasabi na yung metaphase plate doon magla-line up yung mga chromosome. So at this stage, the two kinetochores of each chromosome should be attached to microtubules from opposite spindle poles. So dapat yung kinetochore, yung dalawang kinetochore doon sa centromere ng isang chromosome, doon sa chromatids, it will attach, naka-attach na yung microtubules. So, bale, dalawa yung kinetochore. So, yung microtubules from the north, so let's just say from the north part of the mitotic spindle, mag-a-attach doon sa isang kinetochore. And then yung mitotic spindle from the south part, mag-a-attach ulit doon sa isa pang kinetochore. So, yun yung process na magaganap doon sa metaphase. Lahat ng chromosome ay nahuli na and then lahat ng mga chromosome na yun magla-line up, ready to divide. And then, dapat lahat ng microtubules na nag-bind doon sa kinetochore, lahat ng yun na bind na yung dalawang kinetochore. So ito, yung mangyayari. So, chromosomes will line up at the metaphase plate. So itong metaphase plate na tinatawag, ito yon. Dito lang sa part na to, it is only a term that connotes to the place where the chromosomes will line up. So dito sa part na to, basically dun sa middle ng mitotic spindle. Now, another one is, yun, kasi nag-line up na sila, nahuli na, na-capture na ng mitotic spindle itong mga chromosome, kaya it enables it to line them all up. Now, another one na mangyayari sa metaphase is yung microtubules, itong gray part na to, it will bind to this kinetochore doon sa centromere na dito sa part na to. So, yung north na microtubule magbabind doon sa north part na kinetochore and then yung south na microtubule magbabind doon sa south part na kinetochore. So, yun yung mangyayari sa metaphase. So before proceeding to anaphase, the cell will check to make sure that all the chromosomes are at the metaphase plate with their kinetochores correctly attached to microtubules. So this is called the spindle checkpoint and helps ensure that the sister chromatids will split evenly between the two daughter cells when they separate in the next step. So before daw napupunta doon sa third step, which is yung anaphase, parang may checkpoint na magaganap doon sa metaphase part. So, yung gagawin ngayon ng cell is i-double check niya yung mga chromosome if they are properly aligned doon sa metaphase plate. So, i-double check na kung meron nga bang hindi properly aligned, meron nga bang hindi pa nakapunta doon sa metaphase plate na chromosome. So, yun yung gagawin. And then, another one is, dino-double check rin niya kung yung bang mga microtubules are naka-attach na doon sa kinetochore. So, yun yung i-double check. So, this is called yung spindle checkpoint na tinatawag, which is important kasi para ma-ensure that yung mga sister chromatids, di ba, ito yung isang chromatid, ito yung isang chromatid. So, itong sister chromatid na ito, it will split up evenly. Kasi pag hindi nag-align ng maayos, tapos hindi nag-attach yung microtubules ng maayos dun sa kinetochores, pag split up na ito, posible na hindi maayos, hindi mag-split up into two separate na chromatids. So yun. So if a chromosome is not properly aligned or attached, the cell will halt yung division until the problem is fixed. So yun yung gagawin. Pag hindi properly attached, i-stop niya yung cell division. So, with the alignment of the chromosomes on the plate na tinatawag doon sa metaphase, it is now ready for the next stage or next phase of the mitosis, which is yung anaphase na tinatawag. So, in anaphase, the sister chromatids separates from each other and are pulled towards the opposite ends of the cell. So, the protein glue that holds the sister chromatids together is broken down, allowing them to separate. So, each is now its own chromosome. So, microtubules not attached to the chromosomes elongate and push apart, separating the poles and making the cell longer. So, yun yung mangyayari. So, yung parang importante na mangyari is yung sister chromatids, di ba? Itong mga to dati doon sa metaphase, magkadikit pa lang. Pero sa anaphase, they are pulled towards the opposite ends of the cell. So, yung isang sister chromatid, yung isang chromatid mapupul sa let's just say doon sa south. And then yung isa mapupul doon sa north. So, yun yung gagawin. Ipupul apart nila. So, bakit nga ba mapupul apart na? Kasi yung protein that holds yung sister chromatids together is broken down during anaphase. Kaya, they are able to be separated from each other. So, basically, pag in-split mo or pinol mo apart yung sister chromatids, they will become its own chromosome. So, isang chromosome ito, isang chromosome na ito. So, and then yung mga microtubules that are not attached to the chromosomes elongate and pushed apart, separating the poles and making the cell longer. So, yung mga microtubules daw na hindi naka-attach doon sa chromosome, they will elongate. And with that, they will push apart. Since mag-elongate sila, magpupush apart also sila, and this will separate the poles and making the cell longer. So, yun, as a parang mas zoom in na nung figure. So, microtubules will push or pull apart yung um, uh, spindle, and then yung kinetochore microtubules pull chromosome towards the pole. So yun yung gagawin doon sa anaphase. They will pull apart yung chromatids. So all of these processes are driven by the motor protein, uh, which are yung mga molecular machines that can walk along microtubule tracks and carry a cargo. So basically, yung mga motor proteins, pwede silang pumunta dito sa microtubules, and then they can carry itong chromatids. So yun yung kagagawan mismo noon, hindi mismo yung microtubules, pero it is a combination of the microtubules and itong motor protein. Kasi itong motor protein mismo yung pupunta doon sa microtubule at pupunta doon sa chromatid, at siya yung parang magke-carry ng chromatid. So yun yung sa anaphase. So, after that, after the separation of the chromatids, pupunta na tayo doon sa telophase na tinatawag. So, in telophase, the cell is nearly done dividing, and it starts to reestablish its normal structure as cytokinesis, or yung division of the cell content, takes place. So, the mitotic spindle is broken down into its building blocks, and the two new nuclei form, one for each set of chromosome, and then nuclear membranes and nucleoli appear, and the chromosome begin to decondense and return to their stringy form. So, basically, uh, parang bumabalik sa dati. So, first, sa telophase, di-divide na yung cell into two new cells, kasi 'di ba nag-split up na during anaphase yung mga chromatids. So yung gagawin na is di-divide na ngayon yung cell. And then yung mitotic spindle, na yun yung structure na nabuo noon sa prophase, metaphase, mabobroken down na. So wala nang silbi yung mitotic spindle, kasi nagawa na niya yung part niya, which is to align the chromatids and then separate it. So wala na siyang silbi. So it will be broken down into its building block. And then after that, since parang may dalawa ka ng cell, there will be a nuclei na mabubuo. So, dalawang nuclei yung mabubuo, one for each set of the chromosome. Kasi, di ba, pinol apart mo na yung chromatids. So, basically, may dalawa ka ng chromosome. So, with that, may mabubuo rin na nuclei na dalawa rin in the telophase. So, nuclear membranes and nucleoli also appear. So parang dahil dito parang may new cell ka na nabuo. So the chromosome also begins to decondense. So 'di ba doon sa prophase, sa metaphase, nagko-condense yung mga chromosomes to facilitate yung pag-divide nila. Pero dahil nag-divide na sila dito sa telophase, dahil nag-divide na sila doon sa anaphase, dito sa telophase naman, yung mga chromosomes na yun will begin to decondense and return to their stringy form. Parang ito. Hindi na sila masyadong crowded, hindi na sila masyadong compact. So here is yung parang zoom in view ng figure ng telophase. So the chromosomes will start to decondense. Makikita dito, medyo hindi na sila compact. Yung spindle, yung mitotic spindle will disappear. So wala na yung mga microtubules na naka-attach doon sa chromosome. Then the nuclear membrane reforms. Diba matatandaan na doon sa metaphase, yung nuclear membrane or yung nuclear envelope niya nawawala to facilitate yung pag-align ng chromosome. Pero dito, yung nuclear membrane will reform, and then nucleolus also reappears. Kasi 'di ba doon sa early phases nung mitosis, nawala yung nucleolus. Pero dito sa telophase, dito na siya magre-reappear. Kasi basically parang dalawa ng cell ito. So from one cell due to mitosis, naging dalawa na. So with that, nabanggit ko kanina yung cytokinesis na tinatawag. So the division of the cytoplasm to form two new cells will overlap with the final stage of mitosis. So yun. Pwede, it may start in either yung anaphase or the telophase depending on the cell and finishes shortly after telophase. So basta mag-i-start either dito sa anaphase or telophase at mag-finish siya dito sa telophase. So yung cytokinesis na tinatawag, yun yung division of the cytoplasm. Para makabuo ka ngayon ng dalawang new cell. Doon sa iisang cell, di ba, nag-separate yung mga chromosome. So, bale may dalawa ka ng chromosome. And then, nagbuo rin doon sa telophase ng dalawang nuclei. Nagbuo rin siya ng dalawang nucleolus. And then, meron rin nabuo na cell membrane, nuclear membrane. So, with that, dapat may cytokinesis na magaganap para ma-divide na yung dalawang new cell, para ma-divide na ng tuluyan. So, ito, in animal cell, cytokinesis is contractile. So, it will pinch the cell into two like a coin, pursed with a drawstring. So, yun. Sa cytokinesis dito, diba, isang cell lang to, pero during cytokinesis na, parang ipipinch niya dito sa middle part, na parang coin pursed down with a drawstring. So, the pinch crease is known as the cleavage furrow. Ito. So, yun. So, basically, ito na yung isang new cell, ito na rin yung isang new cell na nanggaling doon sa iisang cell lang during yung the start of the mitosis. So, doon sa plant cells, kasi may pagkaiba yun sa animal cells and doon sa plant cells. Plant cells can be divided like this. Doon sa parang may cleavage furrow ka na tinatawag doon sa animal cells. Because yung cell wall ng plant cells are too stiff. So, medyo hindi masyadong flexible yung cell wall ng plant cells. Kaya hindi ka makakapagbuo ng parang cleavage furrow. Hindi tulad doon sa animal cell. So, instead, there is a structure called the cell plate that will form down the middle of the cell. So, splitting it into two daughter cells separated by a new wall. So, yun. Imbes na parang may cleavage furrow na magaganap, sa plant cell, meron siyang mabubuong cell plate na tinatawag, which will divide yung dalawang daughter cell na nabuo. So yun, with that, after cytokinesis, then you can officially say that the mitosis is done. So basically, PMAT, you have prophase. Doon sa prophase, yung mga chromosome nagko-condense, nagko-compact sila, parang nagdidikit-dikit sila. And then yung mitotic spindle doon sa prophase, mabubuo rin. Now, doon rin sa prophase, nandun rin yung parang pag-attach ng mga microtubules. And then, sa metaphase, dito ka na mag-a-align na ngayon yung mga centromeres that are attached to the yung chromosomes that are attached to the microtubules, so mag-a-align na sila, and then they are parang magre-ready na sila for split, yung pag-split nila. And then dito rin sa metaphase, dito rin mawawala yung nuclear envelope to facilitate yung alignment. And then sa anaphase, dito mag-split yung chromatids into two chromosomes, since mag-split up sila. And then with that, sa telophase, as I've said kanina, dito na mabubuo yung dalawang two daughter cell mo. And then with that, cytokinesis can come When the cytokinesis finishes, we end up with two new cells, each with a complete set of chromosomes identical to those of the mother cell. So, yon. Di ba kanina yung diniscuss ko yung what is definition of mitosis, sinabi ko na yung mitosis, yung goal niya is to parang yung parent cell will divide into two new daughter cells. So, yun yung mga process or steps. So, itong daughter cells na to can now begin their own lives. So, pwede na, depending on what they will decide to. So, kung ano yung gusto ng cell na yon. So, it may grow up to become part of a organ, part of a tissue, or it may undergo again mitosis themselves, wherein it will repeat yung cycle. So, yun yung mga process in mitosis. So, it does not necessarily na lagi nating tatandaan yung mga parang hard na definition or ano. Basta, dapat yung i-memorize lang is yung mga key factors, key processes na nagaganap with each stages of the mitosis. And with that, dapat na if meron kayong hindi naiintindihan, then you could freely go back to the start of the discussion para mamaster ninyo kung ano yung mitosis, kung ano yung mga process na magaganap doon sa mitosis. So with that, let's go naman doon sa isa pa, doon sa meiosis na tinatawag. So ano nga ba yung pagkakaiba ng mitosis at saka yung meiosis na tinatawag? So in meiosis, it is used only for one purpose. In the human body or in the animal, pwede rin basta sa mammals, the production ng gametes, sex cell or sperms, yung sperm or egg cell na tinatawag. So, yun yung meiosis. It is used for the production ng sex cells, yung sperm cell and yung egg cell. So, its goal is to make daughter cells which is exactly half as many chromosome as the starting cell. So, doon sa mitosis, di ba yung gagawin is that it will produce two new daughter cells that are exactly alike with that of its parent cell. Pero dito sa meiosis, yung goal niya ngayon is to make daughter cells that are half lang as many chromosome as the starting cell. So, half lang yung chromosome as the starting cell. So, yun. So, yun yung meiosis. So, to put that another way, meiosis in mammal is a division process that take us from a diploid cell. So, yung diploid na tinatawag is yung with two sets of chromosomes. So, basically may apat ka na chromosome. So, parang ganito. May apat ka na chromosome. Two sets kasi, isang set ito at saka isang set ito. So, yun yung diploid cell. One with two sets of chromosome, to a haploid cell, one with a single set of chromosome. So, maraming parang may mga phases rin siya na pagdadaanan in which magiging ganito na siya. Yung production niya is one single set of chromosome na lang. So, yun yung laging tatandaan in meiosis. It is a division process wherein yung diploid cell, itong part na to, which contains 4 chromosome or 2 sets of chromosomes, yung finished parang end product niya is a daughter cell that contains only one set of the chromosome. So, yun yung meiosis. So, we have, just like on mitosis, on meiosis, meron rin mga phases yan. So, in meiosis, the cell has a more complex task. So, hindi parang straightforward na mga phases ng mitosis. So, it still needs to separate sister chromatids as in mitosis, but it must also separate homologous chromosome. So, the similar but non-identical chromosome pairs an organism receive from its own parents. So, yun. Hindi, parang ise-separate niya pa rin yung sister chromatids katulad nung diniscuss ko dun sa mitosis part. Pero meron rin siyang ise-separate kasi na homologous chromosome na tinatawag. Now, this homologous chromosome na tinatawag, ito yung similar para magkapareho pero hindi mismo na identical na chromosome pairs that an organism receive from its two parents. So yun, i-separate pa niya daw yung homologous chromosome. So, yung homologous pairs separate during a first round of cell division called yung meiosis 1. So, sa meiosis 1, tatandaan na yung homologous pair na nabanggit kanina, magse-separate. Doon sila magse-separate. Then, yung sister chromatids separate during the second round, which is called the meiosis 2. So, yung these goals are accomplished in itong meiosis as a whole using itong two-step division process. Meiosis 1 and then meiosis 2. So, in meiosis 1, di ba, dito magse-separate yung homologous pair or homolog pairs na tinatawag. So, yung homolog pair separates during the first round of cell division. So, sister chromatids separate during a second round. So, makikita dito, before entering or sa start ng meiosis 1, yung cell must first go through yung interphase. So, if hindi mo alam yung interphase, you can go back to the previous lecture kung ano yung interphase. Kasi both cells that undergo during mitosis and meiosis will go into interphase. So, before entering yung meiosis 1, a cell must go through yung interphase. And then, it will go into the process yung mga G1, G2 na process doon sa interphase. So, in meiosis 1, merong tinatawag na Prophase 1 rin sa meiosis 1. So, it differs from the mitosis. Yung mga difference niya from the mitosis will begin to appear. As in mitosis, the chromosomes begin to condense, but in meiosis 1, they also pair up. So, yun yung isang pagkakaiba nila dun sa mitosis part. So, in meiosis 1, particularly in prophase 1 of meiosis, di ba dun sa mitosis, nagko-condense yung mga chromosomes. Pero dito nagpe-pair up sila. So parang ganyan yung nangyayari, nagpe-pair up yung dalawang chromosome. So each chromosome carefully aligns with its homolog partner so that the two match up at corresponding positions along their full length. So parang ganyan, ito yung isang chromosome, ito yung isang chromosome. During meiosis 1, magpe-pair up sila, parang ganito. And then dito sa part na to, sa BC na part ng chromosomes, dito yung mag-e-exchange sila. So it will result into this. So yung pink na chromosome, merong part doon na violet. And then yung violet na chromosome, merong part doon na pink. So yun yung gagawin. They will pair up with each other. So yun, ito yung parang close up. So for instance, inuulit ko yung itong ABC, it will represent yung genes na tinatawag that are found on a particular chromosome, itong ABC na to. And then, the DNA is broken at the same spot on the homolog. So dito, between yung B and C, nagre-reconnect yung dalawang pair ng chromosome. Nagko-connect sila in a criss-cross pattern na tinatawag. So parang ganyan, nag-criss-cross sila so that may exchange na magaganap. May exchange ng DNA na magaganap. So yung with that, yung result na ngayon is yung pink na chromosome, meron siyang part doon na nanggaling doon sa violet na chromosome. Meron siyang DNA na nanggaling doon sa violet na chromosome. Similar to the violet na chromosome, meron rin part doon na nanggaling doon sa pink na chromosome. So, yun yung mangyayari during meiosis 1. So, this process in which homologous chromosome trade parts is called crossing over. So, yun yung process na tinatawag yung pag-trade nila ng parts nila with each other. It is called crossing over. So, the chromosomes would actually be positioned one on top of the other. Yung pinakita kong figure, para lang makita natin, malaman natin ng mas madali kung ano nga ba yung crossing over na tinatawag, yung ano mo ng pag-exchange nila. Pero hindi side by side talaga yung parang positioning ng mga chromosome. They are positioned one on top of the other, parang dito. So after crossing over, the spindle begins to capture the chromosomes and move them towards the center of the cell. So in meiosis 1, dito rin makikita, just like in yung mitosis, yung spindle, yung spindle na tinatawag, huhuliin rin niya itong mga chromosomes na to, katulad doon sa mitosis. And then, ilalagay rin niya doon sa metaphase plate, katulad rin nung nagawa doon sa mitosis. So during metaphase 1, homolog pairs, not individual chromosome, line up at the metaphase plate for separation. So yun yung pagkakaiba nila doon sa mitosis part. Kasi doon sa mitosis part, individual chromosome yung nagla-line up doon sa metaphase plate. Pero dito sa meiosis 1, particularly sa metaphase 1 ng meiosis, pair sila, yung homolog pair na tinatawag natin, yun yung magla-line up doon sa metaphase plate. So, yun yung mangyayari, and then ito yung parang parang phases or phases ang meiosis 1. Katulad rin doon sa mitosis, pero may pagkakaiba lang. So, prophase 1, yung cell mo is diploid. Then, yung homologous chromosome pair up and exchange fragments. So, yun yung magaganap. Sa prophase 1, may crossing over na magaganap. Pero, makikita rin dito na mabubuo rin yung spindle. Meiosis spindle. Then, sa metaphase 1, it will capture yung spindle ngayon, it will also capture yung mga chromosomes. Pero yung pagkaiba nila dun sa mitosis, nasa pair, as a pair sila, that will line up on the metaphase plate. And then, with that, it will go to the anaphase 1. So, parang ganun rin. So, yung homologous separate to opposite end of the cell. So, mag-split up sila katulad rin doon sa mitosis. So, pero yung pagkaiba is that sister chromatids stay together. Kasi nga, di ba, pair sila. Pero dahil pair sila, yung sister chromatids will stay together. Hindi sila mag-split up katulad dun sa mitosis kasi individual chromosome lang yung pumupunta. Kaya nag-split up yung mga sister chromatids. Pero dito hindi. And then, yung telophase 1 ng meiosis, dito na yung mangyayari yung parang sa mitosis rin na may cleavage furrow na magaganap. Meron mga nuclei, mga nucleus na mabubuo. And then, yung mangyayari na is that each chromosome now has two non-identical sister chromatids. So yun, bakit non-identical? Kasi may crossing over na naganap dito sa prophase 1. Kasi yung crossing over, may nag-exchange sila ng let's just say, ng DNA with each other. Kaya, yung mabubuo ngayon doon sa telophase 1 is a chromosome that has two non-identical sister chromatids. So yun. So parang sa meiosis 1, parang yun yung process ng mitosis, pero may mga pagkakaiba lang. So in meiosis 2 naman, it is parang a shorter and simpler process than meiosis 1, and you might find it helpful to think of meiosis 2 as a mitosis for haploid cells. Since, di ba, doon sa meiosis 1, nakabuo ka na ngayon ng from diploid, nagiging haploid cell na lang siya. By the end of the telophase 1 doon sa meiosis 1. Pero dito sa meiosis 2, parang mitosis for haploid cell na yung mabubuo. So yan, uh, it will undergo pa rin yung prophase, metaphase, anaphase, telophase. So parang mitosis na ito, di ba? Kasi may haploid cell ka na, it will line up, then it will be separated doon sa anaphase into two new chromosome, and then just like on the mitosis, it will form itong new cell. So yun yung meiosis 2, parang mitosis ng mga haploid cell. So yun lang yung laging tatandaan, and you will basically know and you will understand kung ano yung meiosis 2 na tinatawag. It is just mitosis for haploid cells. Ito. So with that, that ends my discussion on the cell division, particularly on the meiosis and mitosis. So if you have any questions, you can go back and to the video lecture and look again, or tignan nyo ulit kung ano yung part na hindi nyo na-understand doon sa mitosis and meiosis na part. And you can freely PM me sa messenger, pwede, if you have any clarifications or questions. And with that, that ends my discussion on cell division. So please go over it, please internalize kung ano yung mga sinasabi ko. And if you and I suggest that you can go to additional readings. You can do additional readings, go watch YouTube. Marami dyan mga YouTube videos explaining yung mitosis and meiosis na part. So with that, thank you for listening.